Transportation of gases in between capillaries and alveoli, as well as capillaries and body tissues. Gaseous transport occurs in between the alveoli of lung and heart, and that is called as pulmonary gas transport. And the transportation which occurs in between heart and body tissues or cells is called as systemic exchange of gas. So the systemic exchange of gas transport occurs in between the heart and the cells. The pulmonary gas transport occurs in between lungs and the heart. So the goals for learning here is we'll be looking how the oxygen is transported in blood and how the carbon dioxide is transported, and what are the factors which play a key role in dissociation of oxygen from hemoglobin and favoring the binding of hemoglobin with carbon dioxide. So, coming here, how the oxygen is being transported means, first, the atmospheric air which is being inhaled is being taken into the lungs and they reach the alveoli of lungs. In the alveoli of lungs, the partial pressure of oxygen is high and the partial pressure of carbon dioxide is less, and this favors the hemoglobin to bind with oxygen, forming oxyhemoglobin and dissociating carbon dioxide, which enters into alveoli, and by the process of expiration the CO2 is being released out from the body. So here, who is being playing a key role in the exchange of gases is nothing but the hemoglobin. Hemoglobin, which is present in the RBC cell, is responsible for transporting oxygen and carbon dioxide. The resting body requires 250 ml of oxygen per minute, and we have in our body approximately 4 to 6 billion hemoglobin molecules which are present in red blood cells. So this hemoglobin allows nearly 70 times more oxygen than dissolved plasma. So here the oxygen is being transported by hemoglobin is approximately 98%, whereas in the dissolved plasma it is less than 2%. So the hemoglobin is the pigment which plays a key role in the transportation of gases, oxygen and carbon dioxide. This hemoglobin is a polypeptide chain where it have the 4 polypeptides. They are the 2 alpha and 2 beta chains, and they have the central iron molecule which is responsible for binding with oxygen. So the polypeptides are 2 alpha and 2 beta polypeptide chains. Hemoglobin consists of 4 polypeptide, out of which 2 are alpha and 2 are beta. So each hemoglobin molecules bind with 4 oxygen molecules. So if hemoglobin is bound to 4 oxygen molecules, then we can say that hemoglobin is 100% saturated. If suppose the hemoglobin is bound to only 2 oxygen molecules, then we can say it as the hemoglobin is 50% saturated. Okay, if the hemoglobin is bound to 2 oxygen molecules, then it is 50% saturated. If it is bound to 4 oxygen molecules, then it is 100% saturated. The hemoglobin which is bound to oxygen is called as oxyhemoglobin. So, the hemoglobin which is being dissociating oxygen and binding with carbon dioxide is called as deoxyhemoglobin. So, coming to the hemoglobin saturation, either binding with oxygen and leaving out carbon dioxide, it depends upon the partial pressure of oxygen. When the partial pressure of oxygen is more than that of carbon dioxide, then hemoglobin favors to bind with oxygen, leaving carbon dioxide. So, the oxygen dissociation curves are being showed at different levels. Now, the atmospheric air which is being inhaled will have high partial pressure of oxygen. So, the percentage of saturation of hemoglobin is 100%. Whereas here, as the atmospheric pressure is more, that is the oxygen partial pressure is more, then the percentage of saturation with the hemoglobin is also high. So here it is approximately 100% saturated. Now coming to the transportation, that is from the lungs, the oxygen is transported to the heart, and through the heart the systemic artery collect the oxygenated blood to all the body parts. In the systemic arteries, the partial pressure of oxygen is believed to be 95 mm of Hg. At 95 mm of Hg, the percentage of saturation of hemoglobin is approximately 97%. Now the systemic artery transfer the oxygenated blood to the tissues or cells. At the tissues and cells, the partial pressure of oxygen is 40 mm of Hg. So here the percentage of saturation with hemoglobin is approximately 75%. So out of 75%, that is out of total systemic artery, it is 97%. Out of 97%, 75% is being saturated at the cells or tissue. The difference is 27%, and this 27% is the oxygen which is being transported to the cells or tissues. So the rest of the oxygen is being stored as a reserve in blood. So in the systemic artery, the saturation of oxygen with hemoglobin is 97%. At the cells or tissues, it is 75%. Okay, so here this is the saturation of hemoglobin which is present still in the blood. So what amount of oxygen is being transported to the cells or tissue is, it is only 27%. The rest is being maintained in the blood as a reserve. Now when you consider the skeletal muscles, the partial pressure of oxygen is even more decreased to 20 mm of Hg. So here the partial pressure, as there is drop in the partial pressure, the percentage of saturation is also decreased, and that is even below, that is approximately 35% is saturated. So at rest 100% of oxygen is being dropped off to 75% in the systemic artery. From the systemic artery, it is being transported into the cell where the partial pressure is 40 mm of Hg. So at the exercise or the skeletal muscles, when there is a high fatigue to the skeletal muscles, the partial pressure of oxygen is even more dropped off, that is 20 mm of Hg. At that period, the percentage of saturation of oxygen in hemoglobin is only 35%. Coming to the factors which play a key role in affecting the affinity of hemoglobin with oxygen is well explained by the dissociation curve, that is hemoglobin dissociation curve or oxygen dissociation curve is well explained by Bohr effect. So here, when you plot the partial pressure of oxygen onto the x-axis and percentage of saturation of hemoglobin onto y-axis, so what factors play a key role in dissociation of oxygen from hemoglobin is the partial pressure of carbon dioxide should be more, and there should be high H plus ion. High H plus ion is acidic pH. Acidic pH is less than 7. So the pH should be low. Low pH is acidic, or simply we can call it as a high H plus ion. And there should be more temperature, and there should be high release of 2,3-bis phosphoglycerate by the RBC cell. This is the compound which is being formed by glycolysis of RBC cell, and that favors the dissociation of hemoglobin from oxygen. So, the release of hemoglobin from oxygen or dissociation, breaking the bond between the hemoglobin and oxygen, is being favored by the following factors. That is, there should be more carbon dioxide partial pressure, high acidic pH or increased H plus ions, more temperature, and more secretion of 2,3-bis phosphoglycerate. So here there will be dissociation of hemoglobin with that of oxygen, and the curve is usually moved towards the right side. If suppose the conditions are reversed to this, then it will be moving towards the left side. It favors the binding of hemoglobin with that of oxygen. That is, when the partial pressure of carbon dioxide is low, less H plus ions, decreased temperature and decreased 2,3-bis phosphate favors the binding of hemoglobin with oxygen, then the curve usually moves towards the left side. So, let us consider the hemoglobin saturation at high levels. So, the lungs at sea level, that is the partial pressure of oxygen is 100 mm of Hg, then there is 98% saturation. And when the partial pressure of the lung declines below the sea levels, the hemoglobin still have affinity for oxygen and almost remains fully saturated. So the lungs at a high elevation, that is the partial pressure is 80 mm of Hg. The hemoglobin is again 95% saturated. Okay, even though when the partial pressure of oxygen differs by 20 mm, that mm of Hg, that is 100 to 80 mm of Hg, the hemoglobin shows equal affinity with that of oxygen. Now, at which condition the hemoglobin saturation decreases or the dissociation occurs means when the partial pressure of oxygen is leading to 40 mm of Hg. This usually occurs in cells and tissues, or in the skeletal muscles there is even more drop of partial pressure from 40 mm of Hg to 20 mm of Hg. So this favors high release of oxygen from hemoglobin, or there will be dissociation of oxygen from hemoglobin. Factors affecting hemoglobin saturation are decreased pH. Decreased pH is less than 7, and the temperature should be more, and the partial pressure of carbon dioxide should be high. These are the factors which play a key role in dissociation of oxygen from hemoglobin or favors the binding of hemoglobin with carbon dioxide. So factors affecting the hemoglobin saturation at the time of exercise in skeletal muscle is high temperature and high partial pressure of carbon dioxide. So, the factors affecting the hemoglobin saturation can be reviewed here. They are the blood acidity, nothing but the pH of the blood, the pH of the blood should be low for the dissociation of oxygen, or simply we can say there should be high H plus ions. If the high H plus ions are there, then we can say that the pH is acidic. Or low pH. Low pH means less than 7. The blood temperature, high temperature also favors the dissociation of oxygen. Carbon dioxide concentration, that is the partial pressure of carbon dioxide. So the blood temperature, increased blood temperature from 37 degrees to 40 degrees centigrade usually shifts the curve towards the right side. The blood pH, lowering of blood pH also causes the dissociation of hemoglobin with oxygen, and the curve is moved towards right side. Carbon dioxide concentration also causes the dissociation of hemoglobin with that of oxygen. So the curve is being moved towards right side. So this is the summary of movement of or the dissociation of oxygen and carbon dioxide. In normal condition, the hemoglobin have the affinity with oxygen. When the conditions are being altered, that is increased temperature, high partial pressure of carbon dioxide, 2,3-diphosphoglycerate or bisphosphoglycerate, decreased pH causes the dissociation of oxyhemoglobin curve towards right side. If the conditions are reversed, that is decreased temperature, decreased partial pressure of carbon dioxide, and decreased 2,3-diphosphoglycerate and increased pH, that is more than 7, favors the binding of hemoglobin with oxygen. Formula for calculating the amount of oxygen transported by 100 ml of blood. So, the 100 ml of blood consists of approximately 15 grams of hemoglobin. So, each hemoglobin molecule carry approximately 1.34 ml of oxygen. So, each 100 ml carry 15 grams of hemoglobin. Each hemoglobin carry approximately 1.34 ml of oxygen. 1.34 ml of oxygen. So, the formula here is our atrial oxygen content is equal to the value of oxygen carried by the hemoglobin, that is each molecule of hemoglobin carry 1.34 ml of oxygen. So the value of hemoglobin present in 100 ml of blood is 15 grams into percentage of saturation of oxygen. So here 97% of oxygen is being saturated by hemoglobin. So 97% is 97 by 100 and plus 0.031 into partial pressure of oxygen. So 0.031 is the solubility coefficient of oxygen. So the solubility of oxygen is 0.03 ml and the solubility of carbon dioxide is 0.067 ml per dl. So substituting the values of total oxygen in the blood is 1.34 ml into hemoglobin in 100 ml is 15 grams into percentage of saturation is 97 by 100 plus 0.03 into 100. So if you substitute the values here 1.34 into 15 into percentage of saturation 97 by 100 is 0.97 plus 0.03 into partial pressure of oxygen. Then here it will be 20 ml per dl. So this is how the oxygen is being transported by the hemoglobin. So here approximately 100 ml of blood carry 20 ml of oxygen. So when the blood reaches the tissues, its oxygen concentration is being reduced from 20 ml to 14.4 ml. So from 20 ml, when it is being formed in the lungs, when it is or in the systemic capillaries, when it is reaching the cells or tissues, the value is being reduced to 14.4. So this is the amount of blood still remained, the amount of oxygen still remained in the blood. So how much amount of oxygen is being transferred to cells or tissues means the difference between the atrial oxygen concentration and veins concentration. That is 20 minus 15.4 is approximately 5 ml. So this is the amount of oxygen which is being transported to cells or tissues. Okay, so this is how we calculate the percentage of transportation of oxygen. Now each 100 ml of blood carry approximately 5 ml of oxygen. This is the standard value for calculating the percentage of transportation of oxygen depending upon the partial pressure at atria and at arteries and veins. Now the next is the transportation of carbon dioxide. So the transportation of carbon dioxide is usually happened in three different processes, that is in dissolved stage, or it is being transported in the form of bicarbonate ions, as well as it is being transported by hemoglobin. So here, that in dissolved stage, approximately 7 to 10% of carbon dioxide is being transported. This dissolved carbon dioxide comes out the solution where the partial pressure of carbon dioxide is low. Okay, and this usually happens in the lungs. And there it diffuses out of capillaries into the alveoli, and this is being expired out. So these are the three mechanisms for the transportation of carbon dioxide out, that is in the dissolved stage as bicarbonate ion and carbamino compounds. So here you can see this is the free binding of carbon dioxide with the hemoglobin, as well as formation of bicarbonates. Okay, and it is also released in the form of dissolved stage. So in the form of carbamino compounds, approximately 20 to 25% of oxygen is being carried in the form of carbamino compounds. We have already discussed that this hemoglobin is being bound to oxygen, and this oxygen is usually, or carbon dioxide is usually bound to the amino groups. Polypeptide chains are present to the amino group. The carbon dioxide is being forming carbamino hemoglobin, and this is also formed when the partial pressure of oxygen is less and carbon dioxide is more. Okay, and when the pH is also less and or H plus ion concentration is more, and most of the hemoglobin transports or binds to carbon dioxide in the form of bicarbonates, that is approximately 70% of carbon dioxide is being transported in the form of bicarbonates. Okay. At the tissues, the gaseous exchange, and at the alveoli of lungs, the exchange of gas. Here, at the tissues, you can see the partial pressure of oxygen is less and the partial pressure of carbon dioxide is more. So, this favors the dissociation of oxyhemoglobin to release hemoglobin and oxygen. This oxygen is being utilized or taken up by the cell. In return, the carbon dioxide is being sent from the cells to the RBC cell. Now it reacts with water in the presence of an enzyme carbonic anhydrates to form bicarbonates. These bicarbonates are being dissociated into carbonate ions and H plus ion. So these H plus ions, increased H plus ion concentration is responsible for dissociation of hemoglobin with oxygen. So this hemoglobin now binds with the carbon dioxide to form decarboxyhemoglobin. The bicarbonate ions are now sent out of the cell into the plasma. When the bicarbonate ions are being released out, the chloride ions are being sent into the RBC cell in order to maintain ionic concentration. So how the gaseous exchange occurs at lungs or at the alveoli means the RBC cell now takes the deoxyhemoglobin. Okay, so this decarboxyhemoglobin is being sent into the alveoli. So here at the alveoli of lung, the partial pressure of oxygen is more and the partial pressure of carbon dioxide is low. This favors the dissociation of carbon dioxide and the hemoglobin has a high affinity to bind with oxygen. So here the bicarbonate ions are flowing into the RBC cell. In order to maintain the ionic concentration, the chloride ions are being moving outside. And this is called as reverse chloride shift. Movement of chlorides into the RBC is called as chloride shift. It is called as chloride shift, and moving out of chloride ions in exchange of bicarbonate is called as reverse chloride shift. So here the carbonate ions now interact with the H plus ions to form bicarbonate. This bicarbonate in the presence of an enzyme carbonic anhydrase is dissociated into carbon dioxide and water. This carbon dioxide is now exchanged into alveoli, and from the alveoli by the process of expiration it is being released out. So here this is how there is transportation or exchange of oxygen and oxygen and carbon dioxide with the hemoglobin. So at the tissues the oxygen is being taken by the tissues and carbon dioxide is being bound to hemoglobin because the partial pressure of oxygen is low at cells and partial pressure of carbon dioxide is high. That favors the binding of hemoglobin with carbon dioxide. On to the other end, this carbon dioxide is also transported in the form of bicarbonates we have discussed, and this transportation is approximately 70%. In this way, that is a carbamino hemoglobin, it is approximately 20 to 25%. Only 75% is being transported in dissolved stage. Okay, in dissolved carbon dioxide is only 7%. So these are the three ways how the carbon dioxide is transported. So after collecting the carbon dioxide which is being bound to hemoglobin, it is now going into the alveoli for the exchange or release out of carbon dioxide. When the bicarbonates here are being moving out of the RBC cells, then the chloride ion which is present in the plasma is moving into the RBC cell in order to maintain ionic concentration, and that is called as chloride shift. And in the same way, the chloride ions are going outside when the bicarbonate ions are coming into the RBC cell, and that condition is called as reverse chloride shift. Okay, now here these bicarbonates are being broken down into water and carbon dioxide by the enzyme carbonic anhydrase, and this carbon dioxide is being released into the alveoli. So this transportation is approximately 70%. And here the carbamino hemoglobin, it is with the carbon dioxide which is being bound to hemoglobin is only 20 to 25%. This exchange also occurs at the alveoli, and the dissolved carbon dioxide is also sent into the alveoli. Finally, the carbon dioxide is being released out by the process of expiration. So here, what is the Haldane effect? You know that the increase in carbon dioxide in blood causes the dissociation of hemoglobin with that of oxygen, which we are calling it as Bohr effect. Bohr effect is the dissociation of hemoglobin from that of oxygen. Okay, the reverse of this is also true. That is, when the oxygen with the hemoglobin tends to displace, but that means when they are being breaking their bond with each other, their hemoglobin binds with the oxygen. Okay, binding of oxygen with the hemoglobin tends to displace the carbon dioxide from the blood. So that effect is called as Haldane effect. Okay, oxygen dissociation is from the hemoglobin is Bohr effect. So binding of carbon dioxide is with the hemoglobin is Haldane effect. So combination of oxygen with the hemoglobin causes Hb to be a stronger acid. Thus it displaces the carbon dioxide. Okay. Increase in carbon dioxide in blood causes oxygen to be displaced from hemoglobin. The reverse of this is binding of oxygen tends to displace carbon dioxide from the hemoglobin. Okay, so this is believed that the hemoglobin is acting as a stronger acid. So what is chloride shift means? Moving of chloride ions into the RBC cells is called as chloride shift when the bicarbonate is going into the plasma. So that is called as chloride shift or Hamburger's phenomena. If you like the video, share and subscribe. This is Pravalika signing off.